All right. So let's start with this review session. The goal of this session is really just to, yes, this is all this. The goal of this review session is really to answer any remaining questions about things that we glossed over, weren't clear during this entire week. And then we can talk about other things. So this, I think these two requests are totally along the lines of that. One is when I taught the ML, intro to ML, I skipped over some parts, and we can go and backtrack and look at those parts in more detail. I think in particular, there was a whole part about classification that I just skipped over entirely. So you can go back and go in more detail into that classification bit and just walk through the logic of classification. There is also a last notebook in there that's about, I think, dimensionality reduction, and we didn't get to that one at all. So we can go and look at that one too. And I'll remind myself what's in there because I haven't looked at that in a little while. And then the other request here is great also. This is kind of an extension, just to look a little bit more at a brief overview of Bypi. So I'm of course happy to talk about Bypi at any length, really. But I'll give a short sort of walkthrough of what are the different parts of Bypi. It's a pretty complicated and big library, so I can do that. And then you can feel free to post more things here, and John and I will sort of tag team this. Okay, so maybe we'll start with the machine learning thing. Just make sure. Is there anyone on Zoom? I don't think so. Okay. Well, it's being recorded, and if anyone shows up, they can. Let me close all these notebooks and things from before.
That's a good question.
The question is whether you should download all the notebooks from the server.
And will they lose access to the server after this? No.
Eventually, you will lose access to the server. That's a good question. Eventually, yes, we will have to turn off the server at some point.
It's all on GitHub, so we could make that public.
These materials are all already on GitHub, and you can find all these materials, but I think you'll still be worried the changes that you made to that and so on. And so towards the end of next week and well before this, so A will keep the server running for, I think, at least a couple of weeks after the end of NeuroHackademy. So if you're still working on things with other people and you need more time, that's not a problem. But also we'll give you clear instructions on how to zip up your home folder and download it to your laptop. So it's just a short few commands that you do on the hub, and then you have a copy of your home directory in your laptop, in your own machine. Okay, machine learning, and I think, if I remember correctly, the first thing that I skipped majorly was in here, and that was clustering. Okay, no, this is not the clustering thing that I skipped. Instead, the clustering thing that I skipped. Okay, so putting us back in this tutorial, the first thing that I showed you how to do with scikit-learn is to fit a regression model that predicts the age of participants in the Abide2 data set. I'll remind you again, the Abide data set has 1,004 subjects, and then there are 1,440 brain features quantified from their T1 weighted images using FreeSurfer. And right now I'm running through this first part here, which read in the data. It's doing anything at all? It's not doing anything.
Close this sidebar.
Hmm?
Do this sidebar.
Oh, close this one.
Yeah.
Yeah. No problem.
Ariel, sorry, I cannot I cannot see the slides.
Oh, of course, because I'm not sharing my screen. Thank you. Sorry. I need to get up apparently, because of course I do. 1 second. Okay, here I am.
You're gonna leave yourselves for
There. I'm going to do this as requested, and then kernel, and restart this kernel, and then see that it does what it's supposed to do. Okay, you're just importing stuff. Don't need to think that long. I'm gonna turn it off and then turn it back on again. Discard. Connecting, whatever that means. Okay, I'll really turn it off and then turn it back on again now. Sorry about that. Right, okay, so as I said, we were dealing with this one data set, and the first thing that we did is regression. Or other as having, oh, there it is, it's back. Okay, it worked. Okay, now it's doing the thing. I'm just making sure to walk through everything here so that or to execute through everything here so that I don't miss along the way some variable that I require. Right, so here's the data set, and we're doing age regression or age prediction here. This model doesn't have an intercept now because it wasn't fit, but now it does. And there's the intercept. Okay, good. And these were the results of age regression. And this was other statistics you can do. And this is sort of where we stopped. And so the next thing that we can do here is look at this data set slightly differently. And we'll do, instead of solving a regression problem, now we'll solve a classification problem. Now we could have turned our age regression problem into a classification problem. This is something people will do in the scientific literature quite often, is you have a continuous variable, but you want, for some reason, you want to do a classification problem instead of a regression problem. Maybe you want to compare, you have these younger and these older subjects, and you'd like to compare, I don't know, reading ability in older and younger subjects. And it's just tricky to do when you have this continuous variable. So you discretize your age range into two and compare the younger to the older subjects. Generally speaking, that's almost always a really bad idea. And generally, you shouldn't really be doing that, especially doing classification of older and younger stuff, like taking a continuous variable and doing classification on the median split is not a great idea at all. Instead, what we can do here is try to see whether we can classify different well, try to classify who has autism and who doesn't, who has a diagnosis of autism and who doesn't. And we're gonna start by sort of selecting, not so accidentally, three variables that happen to help with that. So it's they've been selected to correlate with the diagnosis, but we're gonna still use them for the examples. This is not how you would select features in order to do classification, but we're gonna do that anyway here, just for the demonstration, so that we get some results that we can look at and assess the classification procedure. We're going to do use what's called the Gaussian naive Bayes classifier. It's a classifier that uses it's naive in the sense that it assumes that the features are independent of each other and assumes it uses sort of, it's like a little bit like Gaussian mixture model in order to put the classes in spaces like Gaussian distributed blobs. So here we're getting this classifier Gaussian NB, and the really kind of cool thing to notice here is that when fit here to all of the data, X-all contains all 1440 columns. This looks a lot like what we did for regression. That's sort of the important thing, except Y is not a data age, instead it's data group. So it's a, it's got either 1 or 2 in it, right? So I believe 2 is subjects that have a diagnosis of autism. So the Y is a 2-class set of numbers, this binary, and X-all is the same X-all that we used for age regression, and we can go and look at the classification performance of that. So X-all, just to be sure that I understand what X-all here. Yeah, and X-all in this case really does have all of the data in it. So it's got a lot of features to work with here. And in this case, the scoring is the accuracy. That is, you have all the samples. So here we're scoring on the original data. So this is kind of like the overfit case. We're scoring on all of the data, and the answer that this scoring, the question that this scoring answers is what proportion of the predicted values matches the Y value here, the true Y value, right? And the answer to that is 62.5%. So again, one question first to ponder, you might remember that when we did this with regression, and we did this kind of overfit thing, and we predicted age, we were able to predict age with 0 error. And here we're not predicting this with 0 error. So I'm kind of wondering what people think, what's going on here. Why is this not overfit in that sense, that we get every single subject is 100% accurately classified here? I'll give you a hint. This picture is not a bad hint, actually. They're just really mixed together. They really are on all dimensions mixed together. There's not like a way you could carve this thing up. Even in 1440 dimensions, there's no way here you could carve it out so that you get using this kind of classification method. So it's also a limitation of the classifier. I believe this classifier is a linear classifier. It's basically trying to find the surface in 1440 dimensions that cuts between these two clouds these two clouds of subjects, right? But there really is no, even in 1440 dimensions, there's no straight surface that you could put in there. If you're allowed to curve the surface around, maybe, right? But that would be like, in the extreme case, that's like memorizing where each subject is in the space, right? So that's a little iffy. So that's why we get 63, and then you might ask, okay, is 63 a good number overall? And it depends a little bit on what you're trying to do here. If you're trying to very accurately classify individuals with autism, this might not be such a great score. It also doesn't tell you everything that you want to know about the classification performance. But you can, in fact, get more out of classification. There's something called classification report. It's a function that takes then your true Ys and your predicted Ys and gives you this diagram here is helpful. Gives you a full report over all of the subjects. The first thing to note here is that there's a slight class imbalance or some class imbalance between individuals classified as having autism and individuals not classified as having autism. And so the macro sort of accuracy is actually depends on this slight class imbalance. That if you just guessed that everyone in this data set has autism without even seeing the data, you'd already be, 54% accurate. So that's something to take into account when considering that. And then you can drill into this more and ask, among all the possible, so this diagram shows how classification works. You have, your elements, these filled dots, you can think of those as being individuals with autism, and you have your classifier that says all of these people have here have autism. So it's got some of these true positives and some of these true negatives, but it also has false positives and false negatives, and different classification algorithms will sort of carve this space in different ways. And so you can also ask questions like, what is the precision of classification, which is not the same as the accuracy of classification. The accuracy is looking at these that were classified, looking overall here at all of these, what's the match between those that were classified in any of these, either as included inside the circle or outside the circle, and where they should be, right? But precision is really just the true positives over all of those that were classified as having autism. So it answers the question, out of all the people who were classified as having autism, how many do have autism is the precision, and recall is out of all the people who actually do have autism, how many were classified as having autism. And those are two slightly different numbers. The harmonic mean of this number here, is what's called the F1 score. So it sort of takes into account both the precision and the recall. You want an algorithm that has good precision and good recall in both categories here, right? So, for example, if this algorithm tends to have good recall, right? It tends towards having a lot of true positives out of the total number of subjects that do have autism. But for example, the F1 score is slightly lower than that, and it's really differs. The F1 score differs depending on if you are looking to classify individuals who do have autism and individuals who do not have autism. So all that adds like additional subtlety to the way that we interpret classification results. Let's see, what questions do you have about classification overall? So interpreting, the bottom line here is interpreting classification results is complicated. It doesn't boil down to just one number. What's the classification accuracy? In contrast to regression, where often it boils down to like R squared or mean squared error, something like that. Here you need to take into account how are the classes distributed here, and also what am I trying to get out of this? Like, very accurate detection of autism or something else. Right, the control subjects have a higher probability of being correctly labeled, and that has, okay, so 2 here is the control subjects, and they have a higher likelihood of being labeled correctly, among others, because of this class imbalance, and also the nature of the algorithm. Okay, another sort of thing that we skipped over is clustering. So here, this is an unsupervised form of learning, in contrast to classification and regression, which are both supervised learning algorithms, where we had a Y. In regression, we had a Y that was age. In classification, we had a Y that was autism or no autism. In clustering, we don't have a Y to work with. And we're going to here look only at the subjects who are classified as having autism and ask sort of this question about, are there neural subtypes of autism? So that's the kind of question that you can ask with a clustering algorithm. You have some data, and you don't have a priori any reason to necessarily divide this data in any clear way based on the information you have so far. But based on the data, you're asking, is there structure such that this data breaks apart into different subtypes? And that's done. That's called clustering. Here we're going to start with 2 features. So we took out of the data, out of the X all, we're taking only those that have the autism, the data groups that equal to 1. So now Control A, DX1, that shade. There's only the 463 subjects that are classified as having autism. We've also scaled the data. So this, in this case, scaling is quite important. Otherwise, you end up with weird things happening. And so this is the data that we're going to cluster with, and we're going to use an algorithm called k-means. In k-means, we can start out saying we would like there to be, we have to stipulate the number of clusters in advance. And then the way this algorithm works, it's got these 2 features that it's working in. It plops down randomly in the space defined by these 2 features. We'll plot that or scatter that. Right, somewhere here, we're going to plot 4 points. And then we're going to find all the points that are, for every point, we find we call these centroids, the centroid that's closest to that point. And then we're going to call that, I think maybe I have a picture down here. Yes, this is pretty good. We're going to color it according to the centroid. So the centroids here are these things colored in with the darker edge. So we color each point based on the centroid that it's closest to, and then we move the centroid to be the centroid of all the points that were associated with that, and we repeat that process again and again and again until it converges. Yes.
Like forcing 4 clusters versus 6 clusters versus 4 clusters.
Without any justification, it seems like a terrible idea. Like, why would you choose that? And we'll come back to that. But let me actually show the mechanics first. Let's fix K first. We'll deal with that problem in just a second. So you can do this, stipulate the number of features or number of clusters. This should be in advance. And then in this case, we actually run you remember that scikit-learn estimators have a fit method, and we could run fit here and then run predict. But in this case, there's no reason not to run fit and then predict in rapid succession by running the fit and then predict method that does both of those things at the same time. So it takes this data, it runs all this iterative thing that I just described, and then it assigns a cluster for every row in this matrix or subject. And this is very similar to the code we saw before, except there's no y, it's only x. So that's the unsupervised case. And then so here are the clusters that come out of that. Each row gets a number 0, 1, 2, 3, right? Because we posited 4, and then this is the thing that I showed before. These are the two features that were randomly selected in this case, and we're clustering. And so then we can look at this and we can say, well, is this a good clustering, right? What would you use in order to say whether this did a good job or not? I'm curious to hear. Yeah. Yeah, if you had another set in which somehow someone told you, look, there are 4 subtypes of neural subtypes of autism, and we have, I don't know, blood biomarkers on this group of individuals over here, and we know that they are of these 4 subtypes, we could compare this to that. But that turns that into more of a supervised learning problem. Let's just assume for a second we have no idea if there are 4 subtypes or if there are subtypes at all, and we're doing some clustering and we come out with this and we look at this result. How would we assess its quality? Like, is this a good set of clusters? Are these subtypes we believe? Like, what would convince us that these are believable subtypes?
Maybe from a clinical perspective, the symptomatology. If we see some commonalities in the symptomatology between those participants that has been assigned to different clusters. Yeah, so it's similar in a sense to the previous suggestion, in the sense that now we're referencing to some other data. Now maybe we take all the subjects in each one of these clusters and we look at some clinical features and we say, well, look, actually they're if we look at these clinical features over here, they're also separated in their clinical features. So this cluster, and I think that's right. I think that's there's external validation and internal validation. That's called external validation of clustering. External validation is you go to something else and you validate based on that. What would be an internal validation? We only look at this and no other data strategy here. Can we conduct some further statistics, maybe? Like,
what's the data?
Conduct some further statistics, maybe, I don't know, like try to see whether there are some significant differences between the groups. I don't know, I'm trying to think out loud, yeah.
Yeah, there are actually ways to do this. There's so one internal validation method is called Kolinsky-Harabas. It's a criteria. There are actually a number of these criteria. They're implemented in scikit-learn that look at this clustering and look at these points and say things like, you can do something like the Kolinsky-Harabas, if I remember correctly, is what is the compare the sum of squared differences between all these points and their centroid versus the distances between the centroids. Like, things within a cluster should be closer to each other than they are to other things from other clusters. And you get a score, and the better they're separated, right? You can think about, in the extreme case, they're all really, really closely, densely clustered around each one of their centroids, and that's great. But in situations where they're never going to be intermingled, because k-means doesn't allow them to be intermingled, right? They're all, this is a method that carves the space into separate territories. But you can think of these territories being like really close to each other, and a lot of these points being close to the borders of the territories. In that case, this Kolinsky-Harabas score will be lower, and you'll be a little bit more suspicious in internal validation. I should mention one more approach to internal validation, which is something we've seen before, which is cross-validation. You split the data into 2, and then you see whether it replicates roughly. Like, do I get the same clustering if I do it on 2 halves of the data, or is this completely driven by noise in this sample, in which case maybe I don't believe it is much. That's another way to sort of internally validate. Does this thing, when I resample from the data, do I get the same clusters? Right. And then, so, okay, so that's one thing. And then that also maybe answers a little bit of the question about, that you asked about the reasonableness of choosing 4 here, which is that you can, instead of initializing k-means with k, I believe there's a way to tell it to, well, I think if you just do this, it actually goes and calculates some internal validation, tells you how many clusters it thinks there should be. I think it thinks there should be about 8 clusters here. So it does the clustering multiple times based on the features that you gave, and I think it scores it based on something like Kolinsky or Arbaz, and then it chooses one that's the best. And let's see if that will, no, this I think we assumed our palette here doesn't have enough colors. Let's not use palette here. Sorry, I'm going to try to, there we go. So now we get the same colors that we had before, but now you can see it actually thinks this data should have these 8 clusters, and that better represents the data set.
I think actually 8 is just a default.
Oh, it is this the default. Oh God, okay, that's terrible. Sorry about that. There is some way to. Oh yeah, you're right. It's.
I'm double checking.
So I'm just wrong. It is just the default, and this was less. But there are ways to validate the right number, to choose the right number of clusters based on the data itself, based on sort of empirical evaluation. There it is, Kolinsky-Arbaz score. This number is slightly right. The score is defined as a ratio of the sum between cluster dispersion and within cluster dispersion. And so the higher the number, the better. And you can calculate that for different numbers k and see where it peaks. I'll say also you can fit your clusters, you can do even better and fit your clusters to some of the data and then predict some other of the data and calculate there. That would be true cross-validation. And then you can calculate Kolinsky-Arbaz on this left-out data. Yes?
Does that calculation have any like codes or complexity? Because inevitably, as you add more clusters, it will represent data better, smaller and smaller.
Yeah, I think for that, what you need to do is something like cross-validation, right? In the cross-validated case, you actually, I don't think that you would have to have this neat separation between, I might be wrong. Yeah, no, you'd still have to have things fall on this surface. The only penalty I can think about is that the clusters are going to be closer to each other, right? Because there's more and more of them. So they're going to be closer to each other, and you want clusters to be really far away from each other. So that's where you get penalized. Yeah.
I just want to add to that, since I work on the things, and I think we're going to get to dimensionality reduction, but if we use something like a UMAP first, which is like one line to run, then we can specify whether you're more interested in local structure or global structure, and global will generally give you kind of fewer, larger clusters, more broad patterns, and local will be like more small clusters that are more highly similar to one another. It's not a clustering method, but then on top of that, we can cluster or whatever.
Yeah, I'll just add there's like a lot of methods for clustering, and some of them are really complicated and operate on these very high dimensional data. Here we've done this in 2 dimensions, very easy to see. Methods like UMAP and others are really do weird things with very high dimensional data, and it turns a little bit from a science to a bit of an art, and it becomes pretty murky, in my opinion. Right? It gets very murky to work with. So it's a pretty tricky thing to do, clustering in a believable way.
I think even clustering of k-means can be very.
Yeah, even with k-means, yeah. Agreed. Yeah, for all the reasons we saw. Okay. Let me move on to dimensionality reduction. Okay, so this was after everything else. So we're gonna. Right. So we started with sort of, or we got here after doing linear regression and doing regularized regression as well. And when we worked on the regularized regression part of this, the kind of take-home message there was that there are automated methods to gain control of the complexity of your model via this penalized regression approach. One of the things that regularized regression picks up on is the fact that the data may contain redundancies of various sorts, right? So one of the things that will get squashed out when you're doing lasso is if there are 2 columns that are very highly correlated, you don't really need those 2 columns. And if your intention is to use lasso, not ridge, to have as small as possible sum of absolute value of the beta coefficients, it's a good idea to just throw away one of those and really focus on just one if they're very highly correlated, and there's really the differences between them don't help you in solving the regression problem that you're trying to solve. So one way to sort of work with that is to explicitly look for correlations in the data to begin with. So here, for example, we note that the area of left V1, left V2, and left V3 are very strongly correlated to each other. This, I believe, not even normalizing for like overall brain size. So this is because larger people who have larger brains also have larger, I believe, probably has to do with the fact that all their visual areas are also larger. Yes.
Is there a way to put 2 columns that are highly correlated? Is there a way to choose which one to keep, or like to put a guide on which one to keep based on which one might be correlated to a variable that you're primarily really interested in?
Yeah, so there are ways to do sort of feature selection. I think some of what we'll do here is a bit about this kind of feature selection before you do the regression itself, right? So, for example, you might go into before you do you run the algorithm, you say, well, I have a lot of features here, and I don't want to keep all these features. I want to select the features that most closely correlate with this outcome that I'm looking at, age, for example, right? And then you can say, well, let's look at the correlation of each one of these separately, sort them by that, and then just chop off variables that are not highly correlated. Now I have like a much smaller set. I've used the data already to do this. It's a little, need to be careful about at what point I'm doing this, right? I think Russ showed exactly that kind of code where they did feature selection and then went into cross-validation. That has to be part of your cross-validation loop. But if it's in there, you can do that kind of thing. And that's a pretty good way to select features up front if you have lots of features and they're pretty noisy and you're just trying to. In that case, features that are highly correlated and also correlated with your target will still go in together, right? Because they're correlated with each other, they're also correlated with the target. So you're not necessarily getting rid of correlations within the set of features that go into the regularized regression, but the regularized regression itself will tend to sort of select pretty effectively to throw away redundant information.
That makes sense. I guess like, what if we were interested in age primarily and then maybe disease state secondary?
Yeah.
And like maybe these two features, like, you wouldn't throw them out for age, but you would throw one out for disease state.
Yeah.
I don't know, maybe it's just too complex.
There are other ways to take into account, multiple targets at the same time, but yeah, it does get a little bit more complicated. So here is feature selection. In this case, yeah, it's based on F regression is essentially this heuristic that you were just describing, which is let's select the best, 30 in this case, most correlated variables that are most closely correlated with the target. That's F regression essentially. So this is a pipeline that does this. It's again, the pipeline, using this pipeline construct is really helpful because now you can't, when you're doing cross-validation, you can't separate your feature selection from your regression. In this case, the ordinarily squares regression. They're bound together. Or sorry, this is not OLS, this is Ridge. We're doing Ridge with, what's this 100 here? I think alpha is 100 here. Very, very regularized regression. And we can compare that to selecting some random sample of 30 or 300 features instead. And these are slightly complicated. They're learning curves. The idea is to look at the x-axis to see the number of samples that are used for training. Most algorithms will improve with a number of samples, although this doesn't seem to be necessarily the case here for the training in the random features case. It's actually not doing much for us. The 30 best features is not doing much for us versus these random features as well. We disagree with the statement.
Ariel, one question. In the second graph, that would be an
Yeah.
example of an overfitting by using all these 300 random features.
So here, I mean, this is an example of overfitting in the sense that this is higher than that. But I mean, this performance is not so bad. I think the thing to note here, the important kind of thing to notice here is that you can choose 300 random features, and that performs pretty well, 0.6, or you can select 30, best features, and that performs almost as well as selecting 300 random features. So you can either choose, cleverly choose features that are correlated with your target, and then you can do almost as well as if you chose an order of magnitude more features randomly. And if you choose 30 random features, you're down here, much worse. So the idea here is you can reduce the dimensionality. You can do some kind of procedure to reduce the dimensionality of the features that you're using. In this case, the procedure to reduce the dimensionality of the features is by selecting the features most correlated with the target.
And Ariel, one question, and just to be sure if I understood properly one thing. When we were talking about the naive Bayesian, you said that it assumes that the features are independent from each other.
Yeah.
So in this case, for example, that you were talking about the correlation between that strong correlation between some of the features, for example, is that what you were referring to, or is there two different things and I'm just mixing them up?
These are going to be two different things. These 30 best features they may very well be correlated with each other. In fact, they're probably correlated with each other because they're all correlated with this one thing. I'll contrast this with something else. Let's see if we are actually getting to this here. I think we are. So, you know, the contrast that I'll draw here is there are methods that will take your features and decompose them in a way that guarantees that the features that you're selecting are not correlated with each other, right? Here in this case, the only criterion for selection of the features was the fact that they're correlated with the target. And because they're correlated with the target, they may very well be correlated with each other. They're probably correlated with each other, but you can construct your decomposition such that they're, by construction, uncorrelated with each other. So that, for example, one way to do that is principal component analysis. That's actually what principal component analysis does. Principal component analysis, a dimensionality reduction method. The way that it operates is that it, roughly speaking, finds the correlations between all of the variables in a data set, and then it, using those correlations, it finds a way to linearly weight the variables such that you get kind of a variable that accounts for most variance in the data to begin with. That's the first one. And then from the remaining sets of correlations, now you find the next direction that's orthogonal to the first direction, and by construction that accounts for the largest proportion of the remaining data, and so on and so forth until you've exhausted all the variance in the data and constructed all of these PCs. So now you can take and you can replace your features that you originally started with by principal components of the data instead. So you can select, I don't know if exactly that's what we're doing here, but here we're fitting PCA and, oops, interact is not defined. Maybe I failed to import something. Okay. Yeah, this is a interactive plot of this kind of idea. So here we have only 3 dimensions, right? And we see that the one way to think about this principal, the first principal component is that it's a linear combination that defines this first black line here. Linear combination of these 3, actually more of all of the variables, but in this case, we're really only doing the PCA on 3 of the original 1440 dimensions, and explains a lot of the variance. The data is really distributed along this one linear combination. And then the second PC is this orthogonal dimension along which the residual variance, most of the residual variance lies. Here we're doing that with all of this. And so again, like the other unsupervised method, like the clustering method, this has a fit, instead of fit predict, what we're doing is fit transform. So for dimensionality reduction methods, you can think of taking the original data. In PCA, it's relatively straightforward. You started with 1440 dimensions, they're orthogonal to each other, that's the features of the data. Now you've taken and looked at the data and said, well, actually, I'd like my first dimension to be a linear combination of all these dimensions. And the question is, in that, you can think of that as taking your original axes and rotating them around. Now the question is, this point here, where does it sit on that rotated, the first axis of this rotated set of axes? And so what we're doing when we're doing fit transform is we're saying, we're taking the original data and we're moving it to this new space. So now, for example, comps shape, comps.shape will be the size of the original. It's not quite the size of the original data. I'll caveat that in just a second. Original data was 1004 by 1440. So these were the original free surfer features. Now we have instead just 1004 principal components. A principal component analysis is limited. It can't go beyond the smaller dimension of the original data. So we end up with only 1004 features. We've sort of chopped off, I don't know, 400, almost 440 dimensions off the original data. And then, the first one of these is the score associated with right, there's the score associated. These are the scores in the, remember that we did scaling, that's why we get these negative and positive values. We scaled each column in the matrix. So this subject has, in the first feature in the free surfer matrix, they have this value. But in the first PC of the data, they have this value and so on and so forth. So we've moved the data, we've kind of transformed the data around, so we're representing it in this new set of axes. And now one thing that we can do is all of these are by construction uncorrelated with each other, right? Because they're orthogonal. And what we can do now is we can go back to the this feature selection approach that we used before, find the 30 principal components that best correlate with the target, and do regression with those. So that would be a pipeline that includes, I don't think we have it here, but you could do something like dimensionality reduction, PCA, n components equals 30, or n components, we actually don't need n components here at all. We can just take all the components and then, whoops. So now we're doing this F regression selection on PCs. It's having some issues here. I hope it just works. And now, it's even slightly better than this. There's only so much you can squeeze from that, but roughly speaking, that's what it shows. So this is a different way to do this feature selection. You start by doing dimensionality reduction on the data, or reducing the dimensionality of the data. There's more that goes on down here, which is to say, that we can look also at like what is the nature of this principal component analysis. Turns out that a lot of the variance is explained by a first dimension. I'm going to go ahead and just guess that it's related to size of heads somehow. Yeah,
that's actually a question. So I once put one in because I haven't used it that much, but usually it's like being able to look at a PC and then apply data to it and then infer with that PC and then really based on the data running to it. So it turns out like running like free server services or segmentation, how do people do that interpretation? Or just do ROI?
of what the PCs mean, you can pull out of this what are the, I think it's coefficients, components. You can pull out of this set of weights on the original. I don't remember if it's on the rows or the columns though. So there is in here, but I don't remember exactly if it's on the columns or the rows. There is a set of coefficients that tell you this first PC is explained by a linear combination of the following set of regions, and we've weighted, this region this much, this region that much, and that's interpretable. You can take that and you can, if it's ROIs, you can plot it as a map on the brain and say, the first principal component is explained by the following sets of regions, and then the second is by this, and that's pretty well interpreted. Yeah.
Just for the number of components that you choose. I know sometimes they choose just a set number, and sometimes you choose based on explained variance. And I'm wondering if there was a best practice for that.
I think the best practice in this case, we're doing supervised learning. You have like a really good way to do this, which is you do cross-validation. You try increasing number of PCs or something of that sort until R squared starts going down, which is probably what will happen. You'll start by adding the PCA. Generally, you can think of it as also as a denoising algorithm. The initial PCs are a signal from the data, and then at some point, some PCs are just noise. And the question is where to chop that off. In the supervised learning setting, it's very likely that initially you don't have enough variance explained and you need more features. So your R squared is going to start by going up. And then at some point you'll start adding noise and it'll start going down. So PCA in that sense, it's a very nice algorithm to use for in the cross-validation setting, because you can determine the number of PCs based on this kind of U-shaped thing. But if you're just trying to determine how many PCs to use, then yeah, people will often look at this curve and say, look, it's very high. Look, it's gone a little bit lower. It's so low. Oh, it didn't go much lower. Let's stop here, right? They call this like an elbow curve, or there's other names for this kind of approach. And those approaches are also very, very heuristic. Again, you can use a cross-validation approach even in the unsupervised setting, where you split the data into half. You fit PCA to half of the data, and then you try to see whether you can use that in order to accurately reconstruct the other half of the data, right? You apply that PCA to the other half of the data. How accurately am I reconstructing the data in the other half? And use cross-validation in that sense. So there's strategies there as well. Okay, I think actually this is where this is implemented down here. Sorry, I'm not super familiar with this notebook, obviously. And didn't look at it earlier today, so I'm improvising. But yeah, I think this is the pipeline where PCA precedes OLS without the feature selection. You can do this as well, right? You can do PCA and then let ridge regression select the features for you without doing the intermediate F regression step that I did before. And that's actually a really great idea. This is called PC regression. I'm a big fan of this, as you can ask my my students, even in the face of empirical evidence that this doesn't do really well. I'm still very fond of it. And this is running right now. Yes.
This might be a long question, but just in case. I face situations where I'm looking for outliers, or I have like one of the classes is smaller than the other one, and basically I'm interested in small deviations. How does one handle denoising, dimensionality reduction, and things like that when the deviations I'm looking for are rare and probably small, but they should still be larger than the noise? I guess, like in terms of cutting off the number of components.
Yeah, especially for rare kinds of events, like rare, that I'd really worry that if you're doing this kind of approach, that you'd be cutting off the important signal because it just doesn't occupy a lot of variance in the original space, right? It's down, it's down here somewhere. It's not.
Yeah.
So you can't go orderly, PC by PC. Are there other feature selection approaches? So you can do PCA, and then instead of taking like top 30 PCs, you can do the F regression, first of all, right? And that could help in the supervised setting. That could help because maybe there's some PC down here that's really correlated with your distinction of interest, even though it doesn't explain a lot of the variance. And then there are other kinds of subset selections that can help you with that kind of situation, I think. Okay. Well, I think maybe the principle is understood and it's like 4:30. So maybe I'll switch to something else, since I did want to also see what else. Okay, so I did want to touch on Bypi a little bit. So maybe I'll switch over to Bypi now. And then if you have other questions, we can also take those on Slack. So Bypi, okay, I'll go to the Bypi website because that's helpful. Bypi.org. Bypi is the Paragon 3D, 4D plus imaging library in Python. So it says here in the documentation. It's a Python library. It's most of the methods that are implemented here are intended for use with diffusion MRI data, but not only, because the the library includes also several modules that are more general for computational neuroimaging, includes implementations of algorithms, for example, for image registration. And those algorithms were implemented from scratch in Python and in Cython, so that they could be used in the context of as components within more complex algorithms. One way to get started with Bypi is through a set of tutorials, very similar to tutorials you saw in other libraries this week, like NeLearn and Seaborn. There's basically a gallery of these examples. Some of them are sort of very basic things like introduction to basic tracking and getting started, and pre-processing, we call reconstruction, are all algorithms for, and there is a lot of them, for reconstructing the diffusion profile from the signal. These are the algorithms that are at the core of diffusion MRI, because we have the signal and the signal voxel. It's been measured in multiple different directions, and you might want to ask different questions about that signal and sort of reconstruct something about the underlying, the tissue inside of there, the white matter tissue. So one thing you might want to reconstruct are things like the orientation distribution function of diffusion in places in the white matter where multiple different fiber orientations exist. This orientation distribution function might be quite complex. I'll show an example of that. Yes. Here is one of these orientation distribution functions. This is a glyph. Think of this in 3D. We took the signal in a single voxel, and we're trying to model what are all the different orientations of diffusion that are in here. And we're using a method called constrained spherical deconvolution that takes the signal that exists on a, you can think of it as existing on a sphere, and deconvolves that signal with the signal that you would have in a voxel that had only one orientation of one fiber. So deconvolve that with that. And what it tells us really is, you can think of this as weights in a linear regression, right? It says, oh, there's a lot of this single fiber signal over here. And then there's a little bit of that signal over in this direction as well. And if you took all these weights, this is a glyph, so think of it as a three-dimensional function. This is high value here, low value here. Take this and convolve that back with the signal that is generated by a single fiber population, then you would get the signal that was measured in this voxel, or something close to that. So that's reconstruction. In this case, we're trying to reconstruct the orientation distribution function. And this is really useful in tractography. When we come into this voxel using tractography algorithms, and tractography goes step by step, and we follow the diffusion orientations, we get in here, we could say, well, if I came from this direction, there's another sort of peak over here, let me go in that direction. If I came from this direction, I might go here. If I came from this direction, I'll find one of these and I'll sort of follow it. So you can think of tractography as being sort of following these orientation distribution functions from voxel to voxel to voxel, sort of like connecting the dots. And these reconstruction orientation distribution functions serve as the cues for tractography. But as I said, there are many methods for reconstruction, and the purposes of these different methods are slightly different. So here I showed you one constrained spherical deconvolution that's very, very focused on tractography and the orientation distribution function of the underlying fiber structure. But you might want to instead reconstruct something about the microstructure in that particular voxel. I guess this is not a bad one, although I'm not sure that I can explain exactly the model. But you might, instead of inferring something about the orientation distribution function, you might want to infer something about the composition of the voxel in terms of different populations of different kinds of tissue. This is a little bit more phenomenological than that. It's something about the distribution of water spins that make it far away from where they were and then back. And that tells you something about how hard it is for water to move within this particular voxel on average. And how hard depends a little bit on the structure of the tissue within there. Is there a lot of little neurites in there? Or is this just a voxel with lots of space right here. This is the ventricle. Water can go far and come back really easily. But in some other part here, it's not as easy for it to travel far and come back. So those are other kinds of models. And again, there's just a lot of them here, because there are a lot of them in the literature, and different models are good for different kinds of data. It will not surprise you too much to see that one of the things that we do in order to deal with this uncertainty about which model you should use is recommend using cross-validation in order to compare different models. So here we're doing a simple comparison. In one voxel, in the corpus callosum over here on the left, we're comparing two models. I described already this constrained spherical deconvolution, which is trying to find this complicated function, orientation distribution function. The other model that we're using here is one that's a classic, DTI. People will often confuse diffusion MRI with DTI, but DTI is a particular model that was proposed by Basser and colleagues in 1994. And this is a model that reconstructs the diffusion orientation distribution function, as you can think of it as like a sphere or an ellipse or things along the ellipsoid family. It's an ellipse, it's pointing in some direction. So that's the principal diffusion direction. And then it can be very, very selective towards certain directions, or it can be more spherical. And that's this model here, DTI. That was DTI on really a very short amount of time. I can go into a lot more detail about DTI if you'd like. And in this voxel, the idea here is that the method that we're using here is we're fitting the model to some of the data and we're predicting left out data and we're repeating that 10 times. So on the x-axis here is the data, and on the y-axis here is the prediction for every measurement. There are 150 measurements, 150 different directions that were measured. This is a voxel in the corpus callosum. It turns out that modeling the signal in the corpus callosum as this kind of ellipsoid is almost as good in terms of accuracy as modeling it as a combination of weird bumps and blips. But if you go to other places in the white matter, the CSD is actually a better model, having the option to model the fact that there are multiple populations of fibers going there, instead of just either as like an elongated ellipse or as a ball is not as good. So that's one way to deal with the fact that there are all these different models here. Okay, so that was a lot about reconstruction. I'll just say there is also a whole host of methods for fiber tracking that are implemented here. And these reconstruction methods and the fiber tracking methods work hand in hand. You reconstruct the signal or the ODF, and then you pass that on into a tracking algorithm that then can do things either track in a manner that is deterministic. Whenever you come into the voxel, you'll always do the same thing. You'll find the highest peak in the direction that you were coming from and go along that, or it does something a little bit more probabilistic, and there are different ways to do probabilistic things. For example, you might look at the peak and maybe how elongated or non-elongated the diffusion tensor is in that location, and then treat that as like a variance component in a random number generator that you generate a direction from, and then you follow that. So that's one way to generate randomness. And there are other methods as well. And there are details here. There's other ways to sort of use various stopping criteria in tritography. And evaluate, again, this is a method that uses cross-validation in order to evaluate the streamlines instead of evaluating the single voxel models. Once you've done that, you can do streamline analysis, connectivity, analyze tissue properties along the length of bundles that you've extracted. The library includes, as I mentioned, registration methods. So these are image-based registration methods, but also registration methods that apply directly to the streamlines. That's why it was important for us to implement registration methods into the library so that those could be used as building blocks in order to build registration of not only of images, but also of things like objects like streamlines. So there are examples of that, segmentation methods, both in collections of images and in collections of streamlines. And then there's some other kinds of things here. Visualization, you see in a lot of this. A lot of this library creates these kind of nice 3D visualizations that you can use in a lot of contexts. This particular example is kind of the advanced set of visualizations that you can do in order to build little GUIs. So if you enjoy 3D visualization, that can be something to play around with. It's a little complicated. It relies on a separate library called Fury that some of the people working on Bypi is kind of spun off into this entire visualization framework. Okay, that's a big overview of diffusion MRI and Bypi. One thing to sort of notice here, it's not like a specific pipeline that's implemented here. There are command line interfaces that call various components of this, making it easier to run these. But overall, this is a library, and you build applications out of the library, let's say. Okay. Hopefully that answered the request for an overview. Okay. Special considerations for DMRI reproducibility. You want to say something about that?
Sure.
So do you think it'd be a good idea to talk about, for example, like connectivity and endpoints, or like the shapes of tracks versus track profiling, or just in general, like
I guess that is here. Yeah, hey, hello.
Hi.
What are you doing by realizing this?
In my perspective, so I have finally talk about but most of the time that these patients, we have pretty small data set. So I was wondering if advice or things you could consider from that perspective, we want to pretty small data.
Since this is the small data set versus big data set conundrum.
yes, that's a good question. There's definitely benefits to working with small data sets, and I guess when you're working with a small data set, you'd want an algorithm that has a clear signal. So I think I'll just talk about a few of the ways in which people can get into pitfalls with diffusion MRI. Let me see if I can find. I'll go back to Bypi. So I think some of the major, let me see if I can pull up a picture. Actually, let's just use Google image search. Photography. So this is a picture of a tractography. I see these pictures all over the place because I think people think they look very pretty and very cool. I think they look very cool as well. But a lot of people don't know how they're generated. You got an idea of how they're generated when Ariel was talking, when Ariel was looking at a picture of these tensors and describing how you start on one side of a tensor and you're following, you're trying to follow the principal direction of the diffusion signal. Basically, to generate these tractographies, you start somewhere in the brain and you try to follow the underlying diffusion signal to generate what are called streamlines, which are the individual lines in these tractographies. And there's a lot of caveats with these because, as you can imagine, with these voxels, they're often like 2 by 2 by 2 millimeters in volume, which means that inside a given voxel, there could be axons going in multiple different directions. So there's different ways of modeling that. I think the most popular tractography algorithms use a more like, I'll say, liberal approach, where you'll generate streamlines that are both, that some of them are going the correct direction and some of the streamlines, like basically if you have a voxel where there's 2 crossing fibers, you could, and the underlying crossing fibers, there's axons going this way and axons going this way, but you can't distinguish that via the diffusion data. What we normally do would generate some streamlines that go like this, some that go like this, some that go both ways, and some that go like this. And then to make it reliable, you go back and you anatomically validate. What that means is you look for tracks that we already know exist, like the cortical spinal tract, the uncinate, the arcuate fasciculus. There's a lot of large white matter bundles that we already know exist, and so we tend to segment those out of these tractographies. So what are the pitfalls? Some people, it's popular, I think, to do an analysis where you just look at these tractographies without segmenting with anatomical priors, and sort of just looking like, what regions do these streamlines connect, and things like that, especially when you're trying to connect it with functional data. And there's a lot of problems with that, because as I just described, there's this crossing fibers problem, and a lot of these streamlines that you see in this picture probably do not correspond to any underlying axons, just because we don't have enough resolution to follow the axon the whole way through. So you have to validate. Let's go. So let's say, and this is generally the approach our lab takes. So I guess there are some caveats here that different labs might make a different speech here, but this is generally how we try to get the most out of our diffusion data. So let's say you're doing it our way and you've tracked the cortical spinal tract. And it looks something like this. You can start to ask questions about this. Like, for example, where does it start? Where does it end? What's the general shape? What's the width? And a lot of these questions also are not questions that you can really get reliable answers with diffusion MRI data that easily. For example, the diffusion signal inside of the core white matter, where there's a lot of axons, is usually a lot stronger. But when you get to the edges near the gray matter, the diffusion signal there's a lot more, the signal to noise ratio is a lot worse. So asking where these bundles end is a lot harder. So if you have a smaller data set, I think it would be even more difficult. And then also the exact shape and width of this bundle has a lot to do with the exact methodology. So Ariel was talking about how there's different ways of modeling the diffusion signal. He brought up DTI, he brought up CSD. There's a variety of acronyms. If you use different acronyms, if you use those different models, you'll often get different shapes or different sizes for these bundles. And that just has to do with the model you're using. So what can you do? Basically, once you've done the tractography and you've found maybe some bundles that, like, you found some bundles that you've anatomically validated, they're known bundles. Maybe if you have a specific research question, you're only looking at tracks. Sorry, I use bundles and tracks interchangeably. You're only looking at tracks that are specific to your research question. And then you go to that track and you go into where it's sort of in the core of the white matter. So for the cortical spinal tracts, like maybe from here to here, and there are automated tools to do this. And you don't ask a question about the shape of the tracks. You ask a question about the how tissue properties vary along the tract. So you can use another model. Maybe it's the same as you use for tractography. Maybe it's different, but you model the diffusion signal again, and you can get some estimates of maybe like how easily water diffuses, or depending on how complicated the model is, you could get a variety of different tissue properties, but they're basically looking at the diffusion signal and trying to summarize it. I'll show you a picture of what that looks like. Let's see, actually. So in the middle here, you can see an example of what I'm talked about, where they originally generated a large tractography, and then they broke it down into some predefined bundles. So you're only looking at the parts of the tractography that we know exist in the actual human brain. And that helps get rid of all the false positive streamlines you can generate because of the lack of resolution. And then here we walk up to each of these tracks and we don't look at the edges of the tracks. That's what these black bars here on the arcuate fasciculus, the frontal and temporal. We're only looking at the region in between those. And then we sort of walk along the tracks in between those and we ask, what are the tissue properties along the track there? And here we have plotted something called fractional anisotropy. That's basically how anisotropic the diffusion of the water is at those positions. And so it's one-dimensional because it's as you go along the arcuate fasciculus in this case, how does the FA vary? And here he's looking at, it looks like older and younger children and seeing that there's a difference in FA with older children than younger children. So in your specific case, I guess what I would say is try to think about what tracks would be relevant to your research question and then do a methodology. This is AFQ is the exact algorithm we use, but there's a few others like RICO bundles. You can check on the Bypi website and look online. There's a few that use something similar to this, where you're profiling the way known bundles, the properties of known bundles. And I think that will get you the most, that's one of the ways to get the best signal to noise ratio out of the data. So with small data sets, it's pretty important to get something with a good signal. Does that answer the question? Yeah, go ahead.
What is the diffusion signal look like? Is it partial derivatives?
Yeah, Ariel will answer this better, but from my perspective, usually like at a typical data set, you've got a volume where each voxel represents by 2 by 2 by 2 millimeters. And then in each one of those voxels, it's actually 50 different measurements roughly in different directions of how easily did water move along that direction, along that direction, along that direction. And so you've got really 50 different volumes for different directions. And so that's why you take those 50 measurements in a given position, and that's what you're passing into those models like DTI and CSD to try and simplify that, to try and like deal with any noisiness that could arise, but basically summarize all of that data to then use in tractography or to generate these FA things. Anything to add there?
You can also, if it's of interest, I can explain it the whiteboard probably in more detail.
Yeah, sure. Yeah, I don't have anything. Oh, I should, in terms of reliability, another thing to add. So if you, for this hackathon, if you want to work with diffusion data, there's a number of data sets we have that are already pre-processed, but if you want to work with a data set that's not pre-processed, that's okay too. And there's a library we use that is really good for pre-processing diffusion data. It's called QSI Prep. So I just wanted to let you guys know about it if you're interested in it. And then if you want to do the segmenting the tractography into different bundles, that's a library we have called PyFQ, which is, Ariel was showing off Bypi earlier. Most of our library, which does this whole process automatically, is calling off to Bypi. We're just sort of an API around that, where you can just pass in a path to a bids directory, and then we know which Bypi functions to call to get the job done. So you can, that's sort of what our pipeline looks like, but another popular software is this MRTrix. And here they do modeling and the tractography, and I think they have connectomics, which I just said was not like a smart thing to do, but I think they have that in MRTrix. So it's worth checking out, I guess. And are there any other major software things that we should sort of reference if people want to use them?
I guess in FreeSurfer, there's tracking.
Yeah.
The FreeSurfer.
This is another one. Right, any more questions about that? Checking the Slack.
Questions?
There's a question on Slack. Ariel, how has your experience using random forest and SVM models and neuroimaging compared to other models we use today?
Yeah, I mean, yeah, we spend a little time. We work with this kind of data, and one of the things that we try to do is model individual variability in all kinds of cognitive abilities. And so we spent some time in the last few years with Jason Yaten's lab actually looking at reading ability and individual differences in reading ability, and building models that predict who's a better reader, who's higher or lower reading scores. And ultimately, I think I've come sort of to the conclusion that for these kind of brain behavior relationships where the explained variance is not very high, on the order of maybe 10% or so, you don't gain a lot from going from, say, a linear model like a lasso model that similar to the ones that I showed with the free surfer with that Bypi data, to very complicated nonlinear models, even if you have a ton of data. Like a stronger model that maybe takes into account some nonlinearity that exists in there is not going to really boost the performance a lot. But in other domains where maybe explained variance is higher, it could be that it helps a lot. Our recent experience with that is actually in comparing models like Lasso to models like convolutional neural networks that are also complicated nonlinear creatures. It turns out in some cases that doesn't give you a lot. Diffusion MRI doesn't predict a lot of variance in individual differences in things like reading, but it does predict a lot of variance in individual differences in age. So you could predict people's age, the age prediction thing that we did with research. You could do that really, really well with white matter tissue properties in children, adolescents, and aging also. It turns out that even there, you don't gain a lot from using a really complicated convolutional neural network relative to a PCR lasso algorithm. I'm not sure if the lesson generalizes very broadly, though. I will say there are specific cases in which a complicated nonlinear algorithm is just qualitatively different. This is work that John did, where patients with glaucoma, we found in a very large, almost 1,000 subject large data set, where if you look just at the optic radiation of these people and tissue properties of the optic radiation, there it turns out the convolutional neural network can predict with some accuracy, it's not super high, and the linear model does not predict at all. So in some cases, it turns out switching from a nonlinear model to from a linear model to a nonlinear model, some more complicated model is really important. Random forest is a really good kind of first model to run after you've run linear models because it's very generic. It works very well with tabular data with a variety of different kinds. So that's just kind of the common wisdom about random forest algorithms. There's one in particular that we've been using called XGBoost. We're interested in exploring that. It's a little hard to tune because it has a lot of tuning parameters. Like Lasso has one tuning parameter, right? Just how much regularization. This one has all kinds of tuning parameters. So that poses a lot of challenge. You spend a lot of time cross-validating on a variety of different tuning parameters until you can find the right set of tuning parameters. Did that answer the question, more or less? So.
Yes. And just regarding to that last part that you said, so I'm asking because I have already some data, and I had this issue that there are no like components that have like huge variance, like that explain a huge variance. So that's why we haven't tried anything yet. But I'm a bit worried about that. And the first thing that came during our conversation was like, okay, random forest, or SVM should be like the first things that came up, but I don't know if that should be like that, or I don't know. That's why I
Yeah,
was
so
asking.
you also actually asked about this, and I didn't answer. I'm sorry. I meant to post an answer here. You asked a little bit about this
Yeah.
on this channel as well. And I was going to answer that this is a little bit of an interesting thing to maybe simulate and try, right? So the question here was, you know, let's assume for a second that there is some nonlinear relationship between the features and the target. What's the best thing to do if we don't have a lot of data? And my hunch is that up to fairly large amounts of data and low levels of noise, you'd still do better with a linear method than a method that incorporates a lot of nonlinearity, because the complexity of just finding the right tuning parameters for the nonlinearity are going to cost you a lot. But I don't know exactly what that, and I don't know, maybe somebody has quantified these kinds of things before, but it seems like the kind of thing you could simulate. Maybe next week you'd like to try to play around with that, simulate a true nonlinear relationship, and then how much data do you need in order to find that given a certain level of noise. So it all depends a little bit on, unfortunately, on things that are hard to know, like noise. And but my hunch is that the linear methods are going to do really well up to like really large amounts of data and really low levels of noise.
Okay. Yeah, great. Thank you, Ariel.
Yeah, sure thing. Okay, it's 5 p.m. on Friday. I think we'll end here and wish you a nice weekend. Be safe out there, and we'll see you all on Monday at 9 a.m. right here.